Practical 2. Polymerase chain reaction, restriction fragment length polymorphism PCR-RFLP. Polymerase chain reaction PCR. Prepare reagent mixture for PCR by using PCR buffer, DNTP mixtures, magnesium chloride, tag polymerase, forward primer, reverse primer, DNA sample, and distilled water. Prepare a reagent mixture for PCR as stated in the table. Mix the reagent mixtures in a microtube. Vortex to mix the reagents. Set the temperature of the thermocycler as stated in the table. Set the final volume to 20 microliter. Open the lid. Put the microtube samples in the thermocycler. Close the lid. Press start to run the machine. The temperature will increase to 95 degrees Celsius for 10 minutes. Denaturation occurs at 95 degrees Celsius for 30 seconds. Followed by annealing at 56 degrees Celsius for 30 seconds. Then elongation at 72 degrees Celsius for 1 minute. Repeat the step of denaturation, annealing, and elongation for 30 cycles. The last step is performed at 72 degrees Celsius for 8 minutes. Lastly, the temperature is set at 4 degrees Celsius to keep the samples. Open the lid and take out the sample PCR product. Restriction fragment length polymorphism RFLP. Prepare the reagent mixture master mix for RFLP as stated in the table. Distill water. Enzyme. And buffer 10 times. Pipette 2 microliter of the master mix solution into PCR tubes. Add 8 microliter of the DNA PCR product. Mix well by pipetting up and down. Incubate all the tubes at 37 degrees Celsius for at least 2 hours. Mix 2 microliter of loading die with 10 microliter sample. Mix well by pipetting up and down. Samples are ready for the next step.
Agarose gel electrophoresis. Prepare 1.2% agarose by adding 1.2 gram agarose in 100 ml of 1-time TAE buffer. Boil the mixture in a microwave oven for 2 minutes. Make sure the agarose is completely dissolved. Cool down the mixture to room temperature. Pour the mixture into gel gasket. Let the gel polymerize for about 30 minutes. Remove comb from the gel. Transfer the polymerized gel into a gel chamber. Pour 1-time TAE buffer into the chamber. Load 5 microliter of 100 base pair leather into the first well, followed by 7 microliter of sample into the other well. Repeat the step for all samples. Perform electrophoresis at 100 volt for 45 minutes. Remove the gel from electrophoresis tank to a container. Stain the gel with ethidium bromide for 15 minutes and wash with distilled water for 5 minutes on a belly dancer shaker. Visualize the bands using gel documentation system. Gel visualization. Open gel documentation door. Remove the gel from container. Pull out the fluorescent trans UV tray. Place the gel at the center. Close the door. Click Start button on the system. Wait until the process is completed. The result is visualized as bands.